Hey everyone. Today's guest is Dr. Li, who is a postdoc in George Church's lab in the Department of Genetics at Harvard Medical School and at the Wyss Institute for Biologically Inspired Engineering at Harvard University. Today, in hot off-the-presses research, Dr. Li will present findings from her preprint, Human Skin Rejuvenation via mRNA. And with that, welcome Dr. Li, and let's get into the presentation.
Hi, Mike. Thanks for the introduction, and I'm honored to be here to give the talk regarding my recent preprint. So let's go to the science. Hi, everyone. So welcome to the world of human skin rejuvenation. As Mike just introduced, I'm Lily from George Church's group at Harvard Medical School. So as we know, we are experiencing aging every day. So during the increasing of the age, we can see the skin start to have wrinkles and start to be dry, and we have different phenotypes as we can find here. So with the advanced technologies developed in the world, and we are wondering, is that possible for us to rejuvenate skin aging? And how can we start from the old skin and go to the young skin? It's kind of a dream that everyone wants to realize. So as a scientist, we are trying to make that happen. So first, let's have an understanding of the skin aging. So as we can see here, from the age 30 to 40 to 50. So these different phenotypes that we can visually to have this, and we can also know what's undergoing with the microscope. For instance, these epidermis changes and elastin changes, and the word everyone heard a lot of times, collagen changed. So the understanding of photoaging and chronological aging at the molecular level actually is limited. So although that we know these collagens and elastins, they are changed, but we do not know which specific genes they are involved in the aging process. So for us to understand this process, so we search a lot of literature that they published, and we can find that most of them, they focused on the fibroblast, which shows collagens are involved and has limited understanding about the other cells and what will be the driving factors of this aging process. So then that we came up with experimental design with different participants. So for us, so we want to know the whole process about the skin aging. So we started the sample collection from individuals across different ages. So from young age to the middle age to the old age. And also we want to cover both photo aging and chronological aging. So with that, we collected samples from both parts, like the exposure part on the arm, which stands for the photo aging plus the chronological aging, and the samples from the back area, which is only the chronological aging. So after that, we did the single cell iron-seq to know each cells, what's the changes during the aging process. You might ask why single cell analysis? Is that because it's popular these days? Actually, this is just one of the reasons. So another important reason is skin is considered a heterogeneous mix because it's composed of different cell types, like keratinocytes, melanocytes, fibroblasts, and other cell types. So with this, if we treat the tissue and do the bulk iron-seq, so we will mix all of these things together, which make us hard to find which genes actually they play the key role during the aging process. And once that we introduce single cell inside, we can know specific cell types. And each cell types, they will have different behaviors during the aging process. And it's better for us to understand the process of aging and for us to find the key genes that drive the aging process. So for this study that we developed our own skin tissue dissociation protocol, so which that we can capture more cells and more cell types after the clustering. So with this, we will have a better resolution to understand the different cells in skin and for us to develop better treatment. So after this sample collection and introduce the single cell protocols on the tissue, and we did tissue dissociation, and we collected all of these cells based on the single cell study. So we have more than 61,000 cells across different age groups. And we hope that we develop the comprehensive human skin atlas to date and covering both photoaging and chronological aging for us to study. So as we can see here, so this is a single cell atlas. We can treat this as a kind of Google map. So once that we have the Google map, we know from one place to the destination. So we have a specific path. And with this single cell atlas as a kind of aging map for us, we can know from young individuals to the old individuals what's going on. And we can also know like specifically cells, what they play the role inside. For instance, so a lot of studies focus on fibroblasts, and for us, we focus on keratinocytes, which is the controlling like cell type, or we call that the basal stem cell, like skin stem cell for that, to understand like which specific cells to be involved in the aging process. So this Google map, we try to like build like the most comprehensive one, which includes all the cell types in skin, basal cell, spinal cell, granular cell, endothelial cell, also like hair follicles and other cell types. So in our preprint, like we expand the details about all these cell types and their mark genes. So we are like briefly show here for us like each subclusters, we have like the marker genes, and we can find like, for instance, ConquerAging 14, this is marker gene like for the basal cells, and it's clearly like patented in the basal cell population. Then that we might have the question about what's going on about these cell types, right? Once that we have the map, we want to know what's a map for the old cells, what's a map for the like young cells. So this, like we showed like two different agent types, the combined aging, which is photoaging plus chronological aging. And this is the chronological aging. So for the combined aging, we can see from young individual to old to middle to old. And these different percentage of the cells, they are changing. And a clear pattern is the immune cells. And for the chronological aging, because it's like more pure, it's not like photoaging involved. So we can see a clear pattern about the basal cells, and we can also see the immune cells, it increase during the age process. So which match to the current studies, they show that actually aging is related to the immune system. So then that we did a very detailed, like quantitative way to see which specific subtypes they are involved during this process and which will be enriched and which will be depleted. And we found that the skin stem cells, which is like basal stem cells 3, 4, 6, they are mainly depleted during the aging process. It's like in our daily life, we will see older skins, they will have like less elastins and less collagens, and we know like maybe it's related to the stem cell, but we do not have evidence to show that. And for us, our data shows the evidence of the depletion of the skin stem cells will cause the skin aging process. Then that we carefully understand the combined aging and the chronological aging of all of these cell types. So as we can see here, so during the aging process, so these different cell types, their differential expressed genes, they changed, and combined aging, actually, they have more differential expressed gene to be involved compared with the chronological aging. That makes sense because combined aging includes the photoaging part. And it's very interesting for us to find that actually the chronological aging, which is the aging type related to our age that we can see on our ID card, is these differential expressed genes, they normally change for specific cell types. And they have each cell type, they will have a lot of like differential expressed genes. And for the combined aging, that we can see not only these specific DGs, they are also like shared differential express genes. So which means that shared by different cell types. Okay, let's think about what's the difference between the combined aging and the chronological aging. It's a photoaging, right? And so photoaging actually they will cause those cell types to have like connected response. So that means like the UV lights like have the effects on our human skin will cause different cell types to fight together. Then that we validate this our like thoughts like by the Go terms. So we checked these functions of these specific DGs, and we found like for the combined agent, so they are more related to the general phenotypes. And for the photo agents, they will have like specific cell parts like to be involved. So with that, we start to think, okay, we know these like different cell types they changed, and we know human skin stem cells play a big role in the skin aging process. And which is the gene? How can we do that? Then that we start to take a close look of these changes in keratinocytes, which is category includes human skin stem cell. So start with like this structure. So I will give a brief intro to the human skin structure. So this is like young skin, and the outer layer is those like the dead cells that we can see by our hands. And also like so these different cell types, these are the epidermis. So epidermis includes granular cell, spinal cell, and basal cells. And the basal cells are the ones that we call human skin stem cells. So doing this, we can see once the skin aging come to young to the old part. So these like cells, they will have different response. And go to these like gene level. So we did the clustering method based on our in-house algorithm. And we found like 8 groups, 8 clusters in total, and 2 interesting clusters pop up. So one is a cluster that during the aging process, including both combined aging and the chronological aging, these gene expression increased. And another group is during the aging process, the expression is going down. And we found those like clusters, so they have like increased expression. They are like enriched in granular cell, and those like have decreased expressions. They are enriched in the stem cells. And then that we did the network analysis, and we found the hub genes of ATF3. And we measured that the expressions during our studies and other studies to validate ATF3 actually plays the role during the aging process, not only within our data, but also in other data sets. Then once that we figured out ATF3 might be the key gene for us to work with, we start to do the validation project. So the validate is like we start with the siRNA knockdown, which we seed the young keratinocytes. And then that we did the siRNA knockdown, which reduced the expression of the RNA expression of ATF3 in the cells. And afterwards that we measured the beta-GAR, which is a senescent measurement, Ki67, which is cell proliferation, and SASP, which is senescent marker gene. So after that, we can see the results. So this is like the SASP, like the senescent-related marker gene. After knockdown the ATF3, that we can see their expression is increased. So that means ATF3 is related to the cell senescence. And also that we measured the Ki67, the cell proliferation. So after knocking down, these cells started to have less proliferations compared with the normal ones. So it's like mimics the aging process. Once ATF3 expression is going down, the cells are still to have slow proliferation. And we also measured the SA beta GAR, so which is cell senescence status. And we can see at the beginning. So those cells are mostly in blue, so they are not like senescent, but after knockdown the ATF3, that these cells are start to have the senescence level. So with this result that we have already validated, ATF3 in the cells, actually they play the role during the aging process. And then that we come up with our idea, how can we reverse skin aging? Once that we know ATF3 plays a role inside. So as we know, COVID brings MI to everybody. So MI and MI treatment for vaccine for the COVID days gave the public the confidence MI is a great modality and reliable modality to deal with diseases and also like the vaccines. And for us, leverage this knowledge that we build our in-house MI treatment for skin rejuvenation. Start with the synthesis of the MI for ATF3. So we came up with a specific design of the MI that which includes the specific UTRs and the ATF3 genes and also the HbA1 like UTR tails, which like make sure that the expression level is controlled and targeted to skin. So with this MI that we put that onto the older keratinocyte, so to validate the response if like we use this treatment, will that be useful? So we start with like the seeding the older keratinocytes. You can think about like put the older cells there, and then afterwards is apply the MI treatment onto the older cells. After 72 hours, we measure these like index again, the beta-GAR, Ki-67, SASP, and let's see what happened. So here's the result. So we can see like once that we introduce our MR therapy on ATF3, which overexpress ATF3 genes, so the senescent markers that they have significantly down for all these specific marker genes, like for instance, IFN beta, IL-1 beta, IL-6, IL-8. So we can see like several folders changes, and they're both all of them they are significant. And also, were cells to be more? Yes. So cells like start to proliferate more than the control ones with our treatment. So we have like 20% to 25% cell proliferation increase after the treatment. And also the senescent status. So we can see significantly the senescent status changes. And this figure clearly shows that before the treatment, the cells are at the senescent status, and after the treatment, the cells are going back to the young status, which has like reduced the senescent values. And with this, we know, okay, so ATF3 mRNA can reduce the cell senescence, increase the cell proliferation, and how about the fibroblasts? How about the collagens? Then that we start our creative like ideas. So we saw like all these cells within skin, they have communications. Like us, although that we are, in different locations, but we still can be connected by YouTube and by Zoom. So we think like there are ways for us to connect together. So then that we started to study the keratinocyte, fibroblasts, and other cell types of communications. As we can see here on the left panel, all these cells in skin, have like communications and with different range. So the number here is from app like which shows the interaction values of the intensity. So we can see the keratinocyte has strong interactions with endothelial cells, which shows it's related to aging as well. And it has also very strong connections with fibroblast. And which connections are them and how make that? So for that, we gather details, investigation about these like genes to be involved. And we found actually keratinocyte crosstalk, they are like a bulk of like genes, including the collagen 17A1, collagen 7A1, and other cells, other genes. So collagen 17A1 has already been published in many papers. It's a driven gene for skin aging. And fibroblasts, they have these like genes. Once we look at that, you might think, no, it's not related to collagen. actually they are related to collagen very strongly. So A1B1 complex, A11B1 complex, and A2B1 complex. All of these complexes are the collagen integens. So that means collagen will bind on these like complex. So that's actually give us the evidence that keratinocytes have the crosstalk with the fibroblast, and they are linked by these secreted proteins that we list here. And also they will have those like function genes, like for instance, collagens and collagen integen genes play the role in fibroblast. Once that we have these computational results, we start to have our experiment to validate that. So for us, we introduce the experiment. It might be complicated here, but you can think about like there's was like one paper published on the cell cover. It shows they will have the blood exchange from the young mouse and the old mouse. And for us, we introduced that idea to our experimental design. So we will use like the young keratinocyte condition medium. So you can think about it's like part of the medium that we can treat fibrosis. And we will also have like the old keratinocyte medium to treat the fibrosis. And we can see what's the difference between after the treatment, whether young and old keratinocyte medium plays a role on the fibrosis. Let's see. And also like things like we develop the new treatment. So we want to have like the MR treatment, like to have the same effects as the young keratinocytes. So we did this as a control and MR treatment experiment to mimic the young and old experiment. And we want to see if our MR treatment will have the response as the young keratinocyte medium. Then, let's go to the results. So the results show that. So first, let's see like if we treat the fibroblast with young and old keratinocyte medium. So we can see. So with the young keratinocyte medium, actually they have less cell senescence, and they have like four times like of the collagen production. And with the old keratinocyte medium, they have like we normalize it to 1, so it's like the 1-fold of the collagen production. So that means with the young keratinocyte medium treat on the fibroblast, they have like 3 times of collagen production. And what about like our ATF3 treatment? So our ATF3 treatment compares with the control that we did same, and we found like ATF3 treatment also reduced the cell senescence and of fibroblast, and also that we have like 200% of the collagen production. So that means like our ATF3 treatment, not only that we will reduce the stem cell senescence, but also we boosted the collagen production in fibroblast around 200%. So this gives us the confidence and kind of hope that it might happen and it will happen that the MRI treatment for skin rejuvenation. And we summarize that. So we found like keratinocyte plays a big role during the aging process, and it can be the target cell type for us to develop the treatment. And ATF3 that in keratinocytes is a driven gene that drives the skin aging process, and which it will like have connections with these secreted proteins from keratinocyte and have the effects on the collagens in fibroblast. In total, that we bring up this idea and the concept and the results of skin rejuvenation via MRI by ATF3. And in the longer future, we hope it will be a product that we can wrap the MRI into the lipid nanoparticle and put that into the microneedle for the product, and everyone can use this. So then that come to the end. For scientists, we are very excited that we developed this MRI treatment, and we hope like it will be way for us to treat like skin aging and expand to other aging types. And for us, we can conquer like aging, like as Michael's teacher. Yeah, thanks everyone.
That was great. Thanks, Lily. Are you ready for questions?
Yeah, thanks.
Okay, I'm sure there are a couple papers that come to mind. So one of them is where senescent cells were transplanted into the skin. And
Mm.
then not only did that induce localized senescence in the skin, also there were, multiple tissues were uh, had a more pronounced aging phenotype, frailty was increased, cognitive function declined. So did your group look at not just what happened in the skin, but the effect of, the mRNA and the skin rejuvenation? Did you look at the effects systemically in other tissues to see if maybe they were also had positive effects?
Yeah, that's a very good question. So, for us, like initial study is like we focus on the skin aging, and since like skin is attached to the muscles, so now we are expanding to like see the effects on the like muscles. What's the response about our treatment?
So no, so you haven't checked yet to see if that rejuvenated other tissues too.
Yeah,
But just
yeah,
muscle
it's in the process.
but muscle will be the first. Are there any other tissues you're planning on looking at, brain, or even looking at functional measures? Um,
Yeah. So I think like brain will be it's a very good topic. So brain will be the next one, like for the scientific usage, because a lot of like as you just mentioned, they are trying to have like the cell treatment. So inject those cells, like the engineer cells, differentiated like from the iPSCs, like for the cell treatment. I think like for us, like our studies can give them kind of like indicators, like how they can, change the mRNA expressions and to have these like the cell treatment. Yeah.
Nice. All right. So I have questions related to the mRNA expression and like
Sure.
using metabolites. But before I go there, I have another related question to senescent cells potentially having, systemic effects. So,
Mm.
we communicated by email where I sent that paper over where
Yeah.
ATF3 activation leads to reactivation of senescent cell infected virus. So senescent cells that are infected with viruses or retroviruses.
Yeah.
So, yeah, and there can be an increase in viral burden during aging. And with that in mind, so that would suggest that maybe systemically ATF3 is downregulated, and then, to protect the internal, but maybe as a consequence, it gets turned off, in the skin
Yeah.
too. Or is it that maybe the skin is under some kind of viral burden and the skin compensates by turning down ATF3, which then makes me worried if you turn up ATF3 in skin, now do you have, if those effects are systemic, do you have a reactivation of senescent cells infected with, endogenous retroviruses? So what are your thoughts?
Yeah. So first of all, the mRNA design that we created, so it's only targeted to skin. So it will be local like delivery, and it will, even though that we control that, it will only delivered to skin. And think about it, like wildly, it will be, um, incidentally like delivered to other tissues, it will be fine because it will not be expressed because we control the several specific UTRs. It can only be expressed in skin. And I totally hear you that you think about like, these like mechanisms like related to the virus. So will that be a kind of like concerns that we develop these like mRNA treatment for that? And I think for us is we carefully like measures like the response of other genes. So we did the BARCA and seq like afterwards, like to see all of these like gene expressions changes after the MR delivery. So we will try to keep that the safe. It's like not other genes like will be involved and not will treat like other response because of like overexpress one gene. Yeah.
So because it's localized, because your
Yeah.
expression of the mRNA is specifically localized to skin,
Yeah.
there
Yes.
shouldn't be an increase in muscle or, liver, wherever there
Yeah.
may be some viral
Yeah.
problems. Wow, that's fantastic. All right, so then also, um, why focus? So mRNA's got to be translated, right? I mean, granted, you've got, the
Yeah.
what kind of nano, not nano cells, but you're embedding it in
Oh,
nanolipid.
lipid nanoparticle.
Right, lipid
Yeah.
nanoparticles, right? Sorry, I
Yeah.
couldn't, those words couldn't come to my mind. But so that's to get it into the cell. That makes sense. But it still has to get translated, right, into
Mm.
protein and then
Yeah.
all the downstream effects from that. So have you looked into like the metabolomic profile, single cell metabolomic profile to see which metabolites may be responsible? So ATF gets expressed and then which
Yeah.
metabolites in the cell are responsible for the rejuvenation effect. And then maybe you can just, you don't even need a lipid permeable nanoparticle. Maybe just, you've got some metabolites that are uh, soluble. Maybe they're ceramides or whatever they may be. So, what are your thoughts about?
Yes. So once that actually we discovered ATF3 is a driven gene of skin aging. So we developed the mRNA treatment that we put into the preprint. And then we also actually developed like some, as you mentioned, like small molecules of metabolites, specific, like for instance, can that be like, as a kind of treatment, it will be more easy to understand and public will be more easy to accept this modality. So so the answer is yes. So since it's like a talk, as you said, it will be translated into protein, and protein can have small molecules regarding to that. And the reason that we are trying to advocate of MI treatment compared with the small molecule treatment is MI is like more easy to control the expression, and if you use like and also like more effective. So if you use like metabolites or small molecules, so first of all, the effectiveness will be not the same as MI. And the second is, so those like metabolites, actually, they will, even though it will be like, for instance, topping that you can just put on skin, they will like bind to other proteins. So they are not like targeted and precisely. And for us, like we do MI treatment is only ATF3 MI. So it will not be have like only like ATF4 or ATF5, right? So it will be more precise effectiveness and also controllable. For some people, maybe they have like sensitive skin. They are like, metabolites they use, it will like trigger their other response. And for instance, immune response, that's what we do not want to see. So that's a reason for us, even though that we have two modalities to be prepared, we advocate like the MR modality first.
That's a great point, especially if it's one metabolite, does that reproduce the whole effect? Whereas you've got the transcription factor that's
Yes,
initiating all of the, right? It's like you've gone after the biggest tree in the forest rather
yes.
than 50 metabolites that maybe have to induce a similar effect. So what about effects on, so skin pH, so skin acidity, which is a
Mm,
part of the, the barrier,
yeah,
skin barrier function
yeah,
becomes more alkaline during aging. So
yeah.
did you look at the effects of ATF3 on skin barrier, whether it's skin pH improving and becoming more acidic?
Yeah, that's a very nice question. So for us, like we show the data of the skin rejuvenation in the keratinocytes. So keratinocytes is a big part, or let's say majority part of the epidermis. So which controls like the let's say wetness, like these or you just mentioned the pH, these things like on the skin. And it's like another point that I just mentioned. It's like the crosstalk to the fibroblast. So for us, like ATF3, so first, like keeping in mind is our own self, like in the young age, we have the ATF3 like expressed. And then in the old age, it's like they have another amount, just like reduced amount. And for us to, try to control the response is we overexpress that to the young age expression. So we do not like say overexpress like to a huge or like uncontrollable like amount, which so it's just say like at the old age, we're trying to have the young age status. Yeah. So
Uh,
this is how we do that.
yeah, I got all that, but then the effect on like skin barrier function, or is
Yeah.
that first you guys do it in vitro and then it's look at it in vivo like
Yeah.
mice or
Yeah.
something. So have you done any of the animal experiments yet where you've looked at skin barrier function as a result of increasing ATF3?
Yes. So now that we are currently like we are doing two parts. So one is the in vivo like wound healing and UV induced like old mouse, and another part is like we are doing ex vivo skin. So we tested this like on the ex vivo skin, and we can see like the ex vivo skin actually they have the same like index as before and after using the ATF3 treatment. So now that we are getting the data, hopefully that we will have another preprint in the longer future regarding the in vivo
Nice.
data. Yeah.
And then the other aspect of skin is not just the skin barrier function, it's the
Yeah.
skin microbiome. So
Yes.
if you restore ATF3 to, well, not if, but when you restore ATF3 to youthful levels, is
Yeah.
there a change in skin microbiome composition towards a more youthful state? And if so, does that help keep the skin, well, youthful and functional, or more functional relative to older skin? So any plans to look at skin microbiome as a result of increasing ATF3 expression by mRNA?
Yeah, that's a very nice question. That's a very professional one. So actually, after this preprint, there are several groups they want to collaborate. One is like the skin microbiome. So they are very interested in like to see the skin microbiome. I hope this is a plan, and for us, like we will have like more data to show how this treatment like will be changed, or it will, give the evidence like the young age and old age skin, they will have specific microbiome, and with the treatment, what microbiome that will be. Yeah.
Nice.
I'm also very looking forward to see that.
Cool. So then also, why do you think ATF3 is, I mean, it's easy to say just aging ATF3
Yeah.
expression in skin goes down, but
Yeah.
is it in part a consequence of, you've got an increase in inflammation during aging, and then the skin is just a bystander in that process. So that inflammation then leads to a downregulation of ATF3, or do you think that there are more specific mechanisms that the skin is being targeted and ATF3 expression is downregulated? Like, what are your thoughts about why ATF3 would be down with aging in skin?
That's a very good part. Actually, we mentioned part of that in our preprint. So, we have the modules like that I showed, right? So there's the network, and we found like ATF3 is a hub gene. Actually, we know that all these genes like to be connected in that module, and several genes actually downstream is related to the immune system. So that's the reason, like for you, like you ask what might be the mechanism behind that, and for us is like we will say aging that now that we have some kinds of like evidence show it's related to the immune, like the immune cells to be increased and the inflammatory that part to be increased. And for the ATF3, that these connected genes actually shows like ATF3 is exactly the up genes, like upregulated genes, like of these like immune related cell genes. Yeah.
So it sounds like yeah, immune activation during aging is a part of the process that you think why ATF3 and
Yeah.
skin be
Yeah.
down regulated, which
Yeah.
then
I'm like I really like this part because now that actually the follow-up study that we are doing is we are trying to gather like more samples, and we also collect the samples like from acne patients. So this like shows like inflammatory like skin disorders, and we are trying to see if like these like genes, like for instance, ATF3 expression, they change during the inflammatory and what's going on regarding like the aging process and the disease process. It's a very interesting topic. So I really like this, and I hope that we can understand more about that and to understand more about the mechanism part, like how that makes like the age, like skin to be aged. Yeah.
yeah. So what do you think about biomarkers? So if it is immune activation related, that
Yeah.
suggests that you could measure stuff in blood, and that could
Yeah.
kind of be a corollary for maybe, senescent cells in the skin. I mean, you ideally maybe you need a skin biopsy, right? But the skin, I'm sure I mean,
Yeah.
pH of the skin is different everywhere. I mean, armpits, bottom
Yes.
of the feet,
Yes. Yes.
right? So
Yes. Yes.
so, what are your thoughts about biomarkers, like circulating biomarkers, and then linking that to senescent cell burden in the skin and ATF3 expression? And then, if that's the case, if there are biomarkers of it, not just skin related, that suggests that you can modulate the stuff in the blood to potentially slow your rate of skin aging. So, what do you think?
Yes, so actually we have even fancy way. So we are thinking about like the epidermis, like skin um, tape. So blood is kind of like invasive like way, and actually people are like afraid of to do like blood test if they do not have disease, right? Now that we are like collaborating with like one group, so they have the epidermis like tape. So the tape is like kind of tape like put on skin, and then we can have the epidermis data. And then that we build our algorithm to link the epidermis data to our like transcriptome like data and to see the changes of that. And in the longer future, as you said, like we will have these kind of like index like for us to measure that we know what kind of like things like we see in the epigenomics changes, and we can know the how the healthy status of the skin and what we can do. Yeah.
All right, wait, can I ask more about what you mentioned? Like, how would you get basically a measure of the epidermis? I mean, you're just brushing off skin cells, I guess. How is that done?
So it's kind of like specific tape. I'm happy like to send you more information. So the tape like on the skin, and once I put that and you remove that, you can see some cells there, right? So now the technology can do the whole genome like epigenomic test. So after that, we will have these like epigenomic data. And actually the challenge for us to is like to link the epigenomic data to the transcriptome data, right? So, but for them, they have a specific like pool about a lot of like epigenomic data, and they also have some like changes of different genes. And for us, like now we are building the algorithm to link these together, and then afterwards that we can see from the epigenomics change that what will give us like the evidence to see these like derm changes, like other changes of the body. So it's a longer future that I always dream about, like epigenomic clock. So we do not want like everyone to test, like say, oh, you take an image. Image is a traditional way for us to give the index, like how old like the skin is, the person is. And I think like for the bio, like, as you said, like biomarker or bioindex, it will be more precise. And then we can get like the epidermal, like epigenomic data, and then we can know how healthy the skin is. So that's my dream. Yeah.
So this is a side comment, but I used to be 100% go on the epigenetic clocks as great, but when you think about like genomics, and I know you're going to say it's not genomics, it's methylation tags, but
Yeah, yeah.
genomics is just the story of what can happen.
Mm,
Epigenetics for me is starting to move into the same boat where
yeah,
you could have an increased methylation age, but the fact
yeah,
is, what are the actual biomarkers of stuff that's happening, right? So
yeah,
that's proteins and metabolites in the
yeah,
cells.
yeah.
So you could have two discordant data where, the stuff upstream
Yes,
versus
yeah.
right. So I'm not trying to throw water on the epigenetics, but I don't know, maybe they're, metaboloma clock or something else. I don't know, I'm biased. I love metabolomics. But all right, also, so um, wouldn't getting the skin cells, they'd have to, as I mentioned, they'd have to be in multiple sites, right? To see, because I
Mm,
mean, granted, old skin is generally senescent in many different places,
yeah,
probably
yeah, yeah,
ubiquitously, right? But
yeah.
is it the same expression pattern? Is it an ATF3 story in all skin types, all over, right? All over, so
Yes. So I think for us, like we collected the samples from arm and back. So as you said, like skin is very different, like from the face, from the back, from the foot. Specifically, like foot has like very thick skin. And for us, like our data shows is like arm and back. And we have our other data sets, like we showed like in our supplemental figure. So you can see those like the data from the face also show like ATF3 changes and the I think we also got the data like from like other resources, not only the face and arm and back. So happy to like give you more information and feel free to check like the supplemental figure. Yeah.
so the data was the same. So ATF3 expression was down in
Yeah.
all the different skin.
Yeah.
Okay, wow.
Not all like the data that we can get.
Uh,
So yeah, hope like we will get like more data. So yeah, so in the longer future, I hope that we will have like more and more data like for us to have like better understanding. Now it shows like more than like 4 or 5 like locations. The ATF3 is the same. Yeah.
Nice. All right. So then are there, I guess there are, if we fast forward and right, so clinical trials, so you have animal models and then you go to clinical trials, but I also know that the church lab likes to patent things,
Yeah.
right? So
Yes.
are
Yes.
there plans to go to clinical trials or clinical trials and then patent this technology to market it? But and then how long away would we be from, mRNA expression as a way to rejuvenate skin?
Hmm. So first, the future plan. So actually, I think we will do both. Church Lab is very good, like at commercialize these like research, and I think that's the reason I'm in Church Lab. I do hope like our research is not only on paper. I hope like other like specific public they can know this, they can use this. So this is my hope. And the motivation that I'm like excited with this project and working on that for several years. And how long it will like, let's say dream come to our life, right? Now currently, so I'm very optimistic. So we are testing on the ex vivo skin and I consulted with some people, they was in the FDA process for the drug development for skin specific diseases. So they told me like once that we have the ex vivo skin data and then we test on the pig, and afterwards that we can apply for the FDA approval. And also these years, like there are more genomic or gene therapy they are be approved. And for this one, I hope it will be related not only for the aesthetic part, it's only useful for it's not only useful for the aesthetic part, it's also like very useful for those like clinical part. Like for instance, like wound healing, burns, and there are like a lot of like skin disease related parts that we can apply for. It will help a lot of people, specifically like for the wound healing for older population. I have like one advisor actually in our building, so she is strongly support my project because she got a surgery and the wound cannot be healed. So she voted every time, like, I propose this in our institution. She said, like, yes, we knew that for the older population, not only those people they are suffering from severe diseases, but also for the people they have these kind of like concerns, need the help. So we will try to like push forward and try to speed up the process.
All right, so then without, stepping on your own toes, are there things we can do now while we wait for the clinical trials and for the FDA approval to upregulate ATF3 skin expression to minimize, senescent cells in skin and potentially rejuvenate it on our own without the technology? Are there any natural products that can I'm sure you look at all kinds of ingredients in like skincares and you're probably like, that's all, nonsense because you guys can actually change gene expression in skin. So
Yeah,
are there things that can actually, you know,
yeah.
increase?
Yeah, increase. And I think what we are doing right now is like, as I just mentioned, we have two modalities. One is an mRNA and the second is a small molecule. And the small molecule part, like we will also like try to move that faster and to let the public to use or benefit them. And as you said, like the mRNA part, so maybe we will speed up. And a lot of people actually are interested to do the test. They want to be the first one or second one to test the treatment. Yeah, I'm very excited about that.
So as far as you know, there aren't any small molecules that are currently, that aren't, that naturally existing that can upregulate ATF3 expression.
Yes,
Do you
yes,
know of
yes.
any, or do you want to say, or if you say maybe it hurts the patent because then it's like, why do people need to do the mRNA if you can just rub this stuff on your skin now? I don't know which, and maybe you could compare which would be best, right? So,
Exactly, exactly. For us, it's like our institution, we want to cover like most of the modality. It's like ongoing. And I think like I advocate like mRNA because it's more precise, more controllable, more effective. And for the small molecule, it can be like a complementary like part, like for people they do not like this modality or they do have like specific skin concerns. For instance, they do not have like sensitive skin. They want to try these like small molecules. I think it will be like different ways. Like for instance, if we want to have like healthy life, we will do sports and we will eat healthy. So those are different ways actually converge to the same like result, right? And for us, no matter it's mRNA or it's you like metabolites or small molecules, we hope like eventually it will come to the same parts, like let people to have like better looking skin, more confidence, like healthy life. This is our like goal or dream. Yeah.
yeah. So, is there any evidence about like specific diets, like a Western diet versus, I don't know, would pick your diet, Mediterranean or high fiber or carnivore, whatever, pick your diet of choice, influencing skin expression of ATF3? Has anyone looked at that yet?
Um, I haven't looked at that for the 83, but I checked the not specific diet. I checked those pathways actually for the young skin and for the older skin. And specifically, you will be surprised or maybe not surprised at all. Those pathways are related to the metabolism. And also, like as you said, like these like water or these like things like absorption, these parts. So that means actually to maintain good skin is do more sports, eat healthy, and sleep more. So it matched to the epigenomic studies in California. So they published like, one paper, like they have different people from different background, but they have the healthy status like for 3 months. And they check their like epigenomic age. They found that they are like much younger than those ones without like healthy status, healthy like these habits. So Yeah, so I think this part is like very, very interesting. So some part is like I think this is a philosophy. Maybe I took too much. So complicated stuff, I think eventually we'll come to a very simple like part. So it's like philosophy. Philosophy is like simple, complicated, simple, right? So I think it's also for science. So at the beginning, then we think, okay, skin aging is simple, and then we try to make that very complicated to know these like molecular changes or whatever. And eventually, maybe that we end up with a pathways or treatment. It will be another simple way again. So hopefully that, science is also like belong to the philosophy part. Yeah.
Definitely, definitely. All right. Well, that was great. Again, thanks, Dr.
Yeah.
Lee.
Yeah.
And I'm looking forward to see what you guys, produce in terms of research in the upcoming future. So thanks again.
Yeah, thank you so much, and thanks for this opportunity, and very nice for like to talk about the science and share with others. I think I'm really honored to be here. And let me know if you have any questions, and you are very, knowledgeable, like to have those questions and some part that I haven't think about, although they are like talking with me regarding to those perspectives. Yeah.
Yeah, cool. All right. Ciao.
Yeah. Okay, bye, bye, nice. Thanks, Mike.
Yes. Zeit Sie an, Lily.
Yeah,
Bye.
bye.